So welcome to the iwNHL meeting in Bovino in Italy. We're just had our first session and in a very rainy lake region in Italy, however, but I'm very pleased to be welcomed by Anas Younes from New York, Steve Ansell from the Mayo Clinic, and Nathan Fowler from the MD Anderson in Houston, where we have, of course, just been discussing the role of the microenvironment. Hugely interesting topic in the whole of oncology at the moment. Now, of course, the checkpoint inhibitors have got all the press in the solid tumors. And of course, we've had longer preclinical data and stronger data in terms of B-cell malignancies. And of course, you're just presenting data, in fact, that we're seeing much higher responses in some tumors and just as good responses even in the tumors we think aren't responding as well. So first thing is, why has this been an area where hematology, for one of the first times ever, has lagged behind the solid tumor field?
Yeah, I'm not so sure it's lagged behind. I think the attention in the media and in the lay press is a focus on solid tumors for a good reason, because that's where the unmet medical need. So when you see 20% response rate, 25% response rate in lung cancer, where we never thought that it is immune responsive disease, it's a major news. But I agree with you that the highest response rate has been reported in lymphoid malignancies, especially in Hodgkin lymphoma.
Sure. So, of course, in the setting of Hodgkin's disease, we see these high response rates, lots of mechanisms why PD-L1 should be increased on the tumor cells. But, of course, an intriguing feature that you both alluded to is this lack of expression of PD-1 in the microenvironment. How does that reconcile responses to PD-1 blockade?
So I think that's a real challenge, because I think we still have a lot to learn about exactly what PD-1 blockade is doing. I think there are a number of hypotheses. One is that you may be bringing in PD-1 positive cells from outside of the microenvironment, sort of from the peripheral circulation. There may be a direct effect on the malignant cell itself, but possibly you're also just modulating the whole environment, depressing some of the macrophage function, and specifically maybe even changing some of the biology as far as glucose metabolism and other things are concerned. So I think we have a lot to learn. It's always a great thing when you have an effective therapy. Now our challenge is to go back and understand why it's effective.
Sure. Now, Nathan, you raised the issue, and I think in some of your own studies you've been doing it, of the question of the dynamics of things that change and this requirement for serial biopsies. In clinical trials, is that something that patients themselves are willing and to do? What are the logistics of getting regulatory approval to get serial biopsies in?
There are probably many reasons why patients don't, or I guess I'd say patients why biopsies are not performed. Some of that is because of physicians maybe not encouraging them enough, not basically focusing on the importance of these biopsies as part of developing studies or developing science. Clearly, biopsies are uncomfortable for patients and they're expensive. So I think that when we're focusing on biopsies and trying to convince patients to have biopsies, you really have to focus on the benefit to future patients and the benefit to improving understanding about what's going on with some of these new drugs. What I talked about a little bit in today's meeting is that traditionally when we look at biopsies or we look at the effect of different drugs on tumors, we just look at a snapshot in time. And with many of these new drugs, what's happening in the tumor probably changes with therapy. So it's really becoming more and more important to look at these tumors, basically tumor biopsies, as patients get therapy, because they probably change as they get exposed to these new drugs.
So let's start with the diagnostic biopsy. How predictive of response in the lymphomas are these biopsies for the expression of these molecules that we think we're targeting?
So far, I can say, at least from limited information, it is not as predictive as we would like, but the effort's been fragmented. There's no pooled data analysis, let's say, from 200 patients, where we looked with a more objective way. Everyone looked at their 5-6 patients that they treated, and some like in retrospective data, but there's no clue so far that what can predict the response, at least in Hodgkin lymphoma.
So Steve, you were specifically talking about rational combinations. Given that the biopsies themselves aren't predictive in terms of response rates, doesn't that make it much more difficult to plan rationally what the combinations we have to add in to try to augment these responses more?
Yeah, I think it does. And we have a few challenges in that, we don't have a lot of good preclinical biological data, and we don't have a good mouse model. So I think there is a bit of guesswork that has to go into things. I think practically as we begin, we might need to look at different classes and different agents, in particular in different pathways, and base it purely just on efficacy of the therapy in each of those and look at augmenting the benefit by putting those together. As we learn more about the biology, then I think we can go back and say we can actually see ways in which we can take a less active agent but use it to augment a more active agent. I think there is a lot of data to say, and maybe lenalidomide in combination with PD-1 blockade in myeloma, for example, may be a good example of that, where PD-1 blockade on its own in myeloma had very low response rates, but the early data suggests that that combination is very effective. So that's where I think it just exemplifies the complication and difficulty.
Sure. I mean, at least in myeloma, the drug's approved. One of the issues has always been the regulatory authorities require single-agent activity before you get combinations. But what we're talking about here are potential combinations for which there might not be single-agent activity. So what do you think about the notion of how we can be looking from a regulatory point of view in terms of how the companies might develop these going forwards, of trying to put combinations together where you've got potential trials of novel combinations?
Right. So for novel combination now, there's some guidelines put forward by the FDA and I think EMA, and how, what's the mechanism or regulatory path for approval of such. If you don't know the mechanism, which is mainly driven by mechanism, it have to be synergistic and inhibit negative feedback loops or like added value. I think the only way to prove that there is added value of the combination is to randomize trial with 3-arm studies, single agent alone or combination, which makes it more complex. But unless you have a really clear preclinical mechanism that explains synergy, we have to do now this like 3-arm and 4-arm studies, which is doable, but it makes it more difficult to enroll patients.
Sure. I guess, Nathan, that when in this particular setting of, say, myeloma, where you're saying the single agent doesn't have activity, to do that trial would mean potentially enrolling patient onto an arm that you really didn't think was going to have activity. Now, there are creative ways you can do that in terms of closing down arms quickly and having crossover designs that make it more attractive. But those are complex studies to kind of design and execute, right?
They're complex and they take time. And as you know, with when you're designing drug company-sponsored studies, time means a lot. It means a lot when your drug has a certain patent life. And so, what Anas pointed out, I think we're entering a new era where we're probably going to see a lot of these novel combinations. And unfortunately, at least from the way I understand the regulatory approval process requires that you again do a single agent arm and followed by another single agent arm, as well as a novel arm. So at least from what I understand now, we have to do single agent arms in these studies.
But at least they can be done concomitantly. We don't
they can
have
be,
to
and
do the sequential thing
that
anymore.
You could
right?
enroll a very small number of patients. Hopefully, clearly, we would hope to see activity in that arm, but if there's not, we could quickly close that arm and move on to the novel arm as an approval.
So, we talked a lot in the session about, clearly we focused a lot on PD-1, PD-L1, because of course there are agents in clinical trials there. We talked a little bit here about adding in lenalidomide. So what are the other kind of agents that you think are going to be added in that will be specifically looking at targeting the microenvironment? So why don't we
So we've talked about, at least in some solid tumor models in lymphomas, the BTK inhibitor has a favorable, actually on-target, off-target effect, because its ITK is actually on-target effect that may modulate the T-cell response that then make a PD-1 targeted therapy more effective. This is being tested in the context of clinical trials. What's the ideal checkpoint inhibitor drug that it should be combined is up in the air. So there's trials that combine with PD-L1 antibodies, trials that combine with PD-1 antibodies. We don't know which is going to be a winner. I think HDAC inhibitor has been mentioned because it has favorable effect on microenvironment and also modulate PD-1, PD-L1 pathway directly by down-regulating PD-1 and T cells. But there are other, I think it's immune modulators. The question is, how do you balance then safety and efficacy? Because the more we activate the immune response, I think the more we will have an autoimmune reactions and toxicities, and I think that can be seen only in the context of clinical trials.
Sure. Other classes of drugs that you think we could be looking at? Sorry, I know that it's hard to come to after Anas has already taken all the prime candidate.
No, but I think the other thing is that the whole immune checkpoint, using different checkpoints together, is a whole field that's going to take a lot of additional research. So 2 ways you can do it. You can, PD-1 is expressed on cells that are activated and exhausted. So can you really target the more exhausted component and not just the activated component? So can you go after LAG3? Can you go after TIM3? And those are additional markers, or CTLA4s already. All of those are being tested in trials. And then on the flip side, can you actually have a direct activating effect? So can you use an OX40 ligand? Can you use CD27? Could you use some of those molecules to actually, 4-1BB, for example, give a way to actually activate and prevent inhibition all at the same time?
Now, you alluded to this in your talk, this notion of can we reverse exhaustion, or is there a state of exhaustion which is reversible versus getting rid of those cells entirely and bringing in new cells? Which way, in your own mind at the moment, would you think that it's working? Do you think exhaustion of these tumor-infiltrating cells is something that's feasible?
So I think it's a good question because I don't think we have an answer, but I would love to believe that actually exhausted cells are the cells that are being reversed. And the reason I would like to believe that is those cells obviously are tumor-specific. They've become exhausted by repeatedly seeing tumor antigen. So if you now take away the exhaustion, they are tumor-directed and hopefully would have immunological memory. So again, in my mind, the goal is that the patient will remember the tumor cell so that even when in the future, should a tumor cell try and resurrect itself, it would have an immune response that would be directed against it, similar to a vaccine approach.
Sure. And so, Nathan, clearly in lymphomas, you've been leading a lot of lenalidomide studies. Do you think the idea then is potentially once, or if we get lenalidomide approved in this setting, we'll be able to use that as a platform to build many more immune modulatory drugs on top?
I hope so. I think we're in an era now where, unfortunately, the science is kind of lagging behind the clinical data, and we're learning now that ibrutinib, probably drugs like idelalisib, these PI3 kinase inhibitors, and the IMiDs probably have dramatic effects on the microenvironment. Some of these things we're just learning about. So where an IMiD will fit in with these checkpoint inhibitors, I think right now it's kind of unknown because we still need to kind of conduct those studies and hopefully obtain biopsies and to understand what's the mechanism and what's happening within the microenvironment with IMiDs plus PD-1, PD-L1, OX40s, things like that. So the question is, I don't know yet.
Sure. Okay, so with that said, I think you're hearing a lot of enthusiasm in this first session. Of course, I think it's important, the microenvironments where I've focused a lot of my attention, but really good session, got a lot of discussion going, a lot of interest from people in the room, and finally we are seeing that the microenvironment is not just a theoretical target, but really seeing good solid clinical data showing us the importance of the microenvironment in tumours and a real target for improving the outcome for our patients going forward.